Welcome all to this new Gen webinar sponsored by Nanotemper, entitled Improving Large Molecule Production Processes by Integrating Innovative Technology Solutions. I'm Jeff Bogaliskis, Technical Editor for Gen, and I'll be the moderator for today's presentation. The drug development process is no easy road and is often riddled with pitfalls that can send R&D projects spiraling out of control. Protein drugs in particular are a challenging therapeutic area, as biomanufacturers are faced with an array of critical quality attributes and process parameters that need to be accurately analyzed. Protein stability, for instance, is one attribute that requires precise measurements with validated protocols, and researchers are always on the lookout for techniques and instruments that can make the entire process more streamlined. In this webinar, our presenters will tell us about some exciting new process development solutions for monitoring protein stability and how incorporating Nanotemper's Prometheus platform has made those tasks more efficient. Let's meet our speakers, shall we? Marcel Tigges is an Associate Director of Large Molecule Process Development at Janssen Pharmaceuticals in Switzerland, where he leads a team of scientists and process engineers that provides lab operational, computational modeling, and technology transfer support for a broad range of early to late stage products. Dr. Tigges will describe for us some of the production challenges faced by researchers working on large molecule process development and some of the innovative solutions he and his team have devised. Silvia Würtenberger is a product manager at Nanotemper. With her background in protein biochemistry from the Max Planck Institute, Dr. Würtenberger is a great fit to be responsible for the Prometheus product line. Today, she will introduce us to the Prometheus system and how it can aid investigators with their protein stability endeavors. Before we begin, I want to remind the audience to stick around for the Q&A session that will start right after the last talk. Don't be shy and send in any question you might have. It's very easy. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many questions as we can. For now, I'll turn it over to Marcel so we can begin the webinar.
Thanks for the introduction and welcome to today's webinar on new technologies for large molecule production processes. My name is Marcel Tigges. I work in the large molecule process development department of Janssen Pharmaceutical Companies of J&J, and I'm located in Schaffhausen, Switzerland. You also see here a little snapshot of the site where we have end-to-end cross-functional capabilities to support the parenteral fill-finish operations. A short disclaimer on the content of today's slide deck for your acknowledgement. And the agenda of today, with the goal to give you a kind of top-level overview of the fill-finish of parenteral products and process development, some insight into CQA, CPP controlled process optimization design, so CQA being critical quality attributes and CPP being critical process parameters. Two examples also very briefly described for exemplary data, with the nanoDSF allowing for a low volume assessment of protein stability and aggregation behavior, and the FlowVPE in the framework of PAT for large molecules, so process analytical technologies, to better monitor the protein concentration in real time and inline. DPDS Schaffhausen is a local center of excellence as part of a global R&D organization, which is basically spanning all over the globe. We have in Schaffhausen all the surrounding functions like AD, so analytical development, DPD, drug product development, the clinical supply chain with the different production scales, but also the small molecule value stream in close proximity. On top, we have in Schaffhausen the commercial production side of JSC, so the Janssen supply chain, and all together that allows us to really look at the entire range of production scales, drive a lot of cross-functional efforts, have the scale up, scale down capabilities, and in general provide end-to-end support for basically any NME or lifecycle management product. You also see here some representative numbers of potential batch sizes at the different scales. So really ranging from the small liter or even milliliter range in the lab up to the commercial, where we talk about 1,000+ liters. A short refresher on the large molecule manufacturing process. I guess most of you are aware of at least parts of it. It all starts with the cells and raw materials, so really transfect your production cell line with the respective target, generate a cell bank, go to the small scale cell culture, scale it up to large scale cell culture for higher yield production, recover your product, filter your product, formulate your product, pool this formulated bulk, fill it into the primary container, which could be either a vial but also a syringe, lyophilize it or keep it at the liquid representation, stopper and cap your respective container, inspect the product, label the product, package the product, and then ship it to the respective recipient. The fill-finish operation in this roadmap here is sometimes underestimated with respect to the complexity and also potential impact on the drug product. And that's why one major task also of my function within the drug product development is to, let's say, ensure a flawless execution of the fill-finish activity to guarantee highest quality at highest yield in shortest turnaround. Zooming in now into the Parenterals fill-finish process flow, we can use this little scheme here to identify 3 major unit operations that need to be tightly controlled. So one is the compounding, the other one filtration, and then the filling. Of course, there's additional steps, but let's now, for the moment, have these 3 as major unit operations. Each of these steps is tightly controlled by the so-called IPCs, so in-process controls, which are also part of the batch record. Process controls here highlighted in blue that allow us to even better understand and optimize the process, and then general monitoring, like also environmental monitoring tools. You see there is a lot of relatively basic tests like pH and osmolality, that can be done in relatively short turnaround times, but there's also others like bioburden, endotoxin or protein concentration, where the offline testing is, let's say, of the essence to continue with the process. So we would implement hold times that obviously block the production from continuing to the next unit operation. So the overall goal of the implementation of new technologies is on the one hand not only to increase, let's say, the quality of our analytical readouts, but on the other hand also by replacing offline testing to massively shorten hold times while maintaining or even increasing quality, and getting maybe insights into additional drug product or process attributes. To support now a flawless execution of these unit operations that I outlined on the slide before, we carry out different studies in the lab. The slide here shows the studies we do for mixing, either in disposable systems, here shown with the white carriers carrying disposable bags, or in stainless steel tanks of different sizes. The goal of DPD in this perspective is to evaluate the optimal mixer settings according to a respective vessel size. This is mainly looking at the speed of the stirrer, therefore providing the correct RPM to production at scale. The second is homogeneity of our drug product and blend times. We also look at potential drug product impact and process limits, mainly by shear forces generated during mixing. There are also additional phenomena like foaming and degassing, which should be avoided since they either reduce the yield or the quality of our drug product. We perform these mixing studies already with the implementation of PAT tools. Here already a little picture of the FlowVPE that would allow to continuously monitor the protein concentration during mixing and support this experimental data then with modeling data generated in this example here with Mixit. You will learn more on the protein concentration measurement then in the, let's say, more in-depth technology part later on. The second unit operation that we support with lab studies is the filtration. Our main areas there is the Vmax studies to select the right filter and scalar properties. We look also at pressure impact studies, which is a regulatory requirement, and the protein and surfactant absorption at the filter, also using PAT tools, in this case again the FlowVPE for the protein concentration. You can imagine that any absorption of drug product to the filter will significantly reduce your concentration in the respective vial. So it is of the essence to get a good idea from which time point on or which volume on you have the target protein concentration. In the lab we can mimic various setups, either with different pressures, different volumes, or applying completely different architectures of the filtration. The third unit operation that we routinely support with lab studies is the drug product filling itself. There we have in general 3 different pump types, so the peristaltic pump system, the rotary pump system, and the time pressure system. The studies are all focusing on fill accuracy, potential drug product impact, and generating fill curves and parameters that then can be transferred to larger scales. The major monitoring there is if you have a kind of homogeneous fill according to the needle movement, the tubing length, and the speed of the pump, to avoid what you also see there in the 3 little pictures on the lower right, and to avoid then, for instance, air bubbles or splashing that would cause underfills. Our experimental studies are supported by numeric modeling, and here's a kind of a glance on 3 different options that we carry out on a day-to-day basis with our in-house modeling group. So one is the pure statistical models like factorial and surface response DOEs, MVA, PCA, and PLS analysis, the mechanistic models like simplified process balances, full computational fluid dynamics modeling, or the classical engineering approach like pure empirical correlations, also based on historical data, dimension analysis, or similarity. So having now a short look on the drivers behind the process optimization, we can identify 2 major areas. One is coming from the drug product itself, so the CQAs. One is coming from the process, the CPPs. This could be API production, the fill finish itself, but also simple things like packaging or shipment. Some of these attributes are pretty generic. So if we look at particulate matter, pH, or the product concentration, you will find them obviously as CQAs basically in any analysis. If you align them with the specific process, you might identify areas that are very harmful to the product. So they would form additional problems, or let's say cause additional stress, while others would remove this stress. The goal of a criticality analysis is to align the product-specific CQAs with the process-specific CPPs to be able to generate or identify the best control strategy and eliminate stages or process steps that are very harmful to your product. This is also a bit outlined here. As said, this is the so-called control strategy. So you identify the CQAs and the CPPs and the CMAs. CMAs is a new abbreviation, sorry for that. That's now the critical material attributes, since also silicone tubing, leachables, and extractables can cause a lot of impact then on your drug product, especially when putting it on stability for the shelf life assessment. So all these together are part of the risk assessment and then the design of the effective control strategy, which is continuously monitored and optimized over the lifetime or the life cycle of your product. Summing up now a bit this very brief introduction, and please don't hesitate to ask questions if something was a bit too brief and a bit too short, but for the sake of time I just try to give an overview. So summing this now up with the major challenges that we face in our daily work, we clearly can identify that we basically always have limited material, especially for NMEs. API production is not meant to go to a large scale. Second is the turnaround times for analytical testing, which is also very intuitive since most of the methods need basically, again, quite some material to get to a validated stage. In the early stage of the product, you still have frequent changes and adaptations, which could be as little as, let's say, adding or changing the concentration, or as massive as changing the formulation. The stability sampling in combination with accelerated timelines poses a huge challenge to our lab work, since stability obviously is always over time and you cannot really cut there. In principle, you always do accelerated for 1 month at 40C. That gives you some indication, but the real stability data comes then really from a longer term storage for several months. And this is obviously massively impacting then our turnaround times. An ever-occurring challenge is the scale-up itself. Not all of our tests can be directly implemented then at commercial scale or also a larger clinical scale. So we are continuously optimizing this challenge or this problem. We have the comparability issues, meaning you have batch-to-batch variability, you have sometimes different methods carried out at different sites, and due to the increasing amount of outsourcing, you also need to always take into account how is the process carried out at a CRO versus your internal procedures. And the root cause identification, which is a bit of like playing Sherlock Holmes. At the moment where you identify an issue, you have very short time to really identify then the reason or the root cause for that. So with all these challenges, we are constantly looking for new technologies that allow us to, let's say, use lower volumes, high throughput and or real-time analysis of these CQAs and CPPs towards being able to continuously monitor this control strategy. And the 2 examples I will show you now in the next like 10 to 15 minutes is, as promised, the protein concentration in real time, FlowVPE, and a new tool to better monitor shear stress with the nanoDSF. So let's now have a look at these 2 tools. One, as said, the FlowVPE, and the other one, the nanoDSF. We start with the FlowVPE. So this is the situation before implementing the FlowVPE. You can now imagine a scenario where you receive the frozen formulated bulk at a certain concentration, like 10 mg per ml. You pool and mix this formulated bulk. You need to take the first IPC, so in process control for protein concentration. You measure that offline in the lab. You feed back the result to the production with a turnaround time of something like ideally 2 hours, at some CMOs even 24 hours, which would require then a hold time of this respective time frame here. You then add your buffer to dilute it down to the target concentration, in this case of 2 mg per ml of the drug product. You would take another IPC to verify the protein concentration again offline. So you can imagine that this is suboptimal, not only for the hold time, but also for, let's say, getting decent real-time data of your dilution process. So which options do we now have to, let's say, move on to the next generation of protein concentration measurement? If we look at the classic A280 gold standard method, I think we all know the Lambert-Bear's law, where you calculate the concentration out of the absorption at 280 nanometer and the extinction coefficient of the respective molecule. C Technologies developed initially the SoloVPE that is still relying on the Bear's law, but now replacing the single absorption measurement by a slope value that is measuring the absorption at different path lengths. This slope is then used to calculate the concentration together with the extinction coefficient. This technology has quite some benefits over the classical method, so we no longer need a dilution in the range of something like 0.5 mg per ml of the protein up to 200 mg per ml of the protein. We don't need a buffer blank, and this technology has also the potential to work inline. This is exactly what is then, let's say, the latest stage of the variable path length measurement, namely the FlowVPE, where you just plug in this tool here, so this flow cell, into your process. So you would have here the inlet of a tubing and the outlet of the tubing. You would have a flow cell, a moving optical fibrate, which would vary the path lengths, and by that you could then really in the process measure your protein concentration. Again, major benefit, real time, no dilution. The way Janssen has implemented that into its process is, on the one hand, for the mixing and compounding. You see here the recirculation loop with a peristaltic pump, the flow cell in this loop going back then to the mixing tank. You can now imagine that with ongoing dilution and this recirculation, you get a very decent readout of the concentration values. Another option that is possible with the FlowVPE is the vial filling itself, so that you basically get 100% control of the protein concentration in any vial that you fill. Looking at some data that has been generated with the tools, we can maybe start with the mixing and compounding. So again, the setup where you have your mixing tank, the recirculation loop, the FlowVPE within this loop going back to the mixing tank. If you now have here on the x-axis the time and on the y-axis the concentration, I hope you can read. It's pretty small, but I think the message is still clearly visible. You would see that at a starting concentration of something like 2.5, you would take an IPC, this is the green dot measured by the classical A280, and you would in parallel measure the concentration in real time, which is the black dotted line representing the FlowVPE data. If you now start at 2.5, you add the API, you can now in real time follow your concentration until the target of something like 7 mg per ml. In former times, you would need to take individual samples and also take into account at which time you take these samples to conclude on complete mixing. This is now eliminated by just following the protein concentration in real time. The second option there would be if you now not add the API, but if you just dilute your drug substance. This would be basically a bit of the inverted scenario. The setup is the same. You would again mix, you would add your buffer, and you could again nicely follow what happens with your protein concentration over time to decide then on the endpoint of your dilution. As said, for the vial filling, a very nice feature would be to see if every vial receives the target protein concentration. This is outlined here. You see that from time 0 on, your protein concentration is within the spec, so you have an absolutely 100% control of filling. Additionally, if you now have a filter in front of your vial filling, you could also nicely detect the filter absorption, which is outlined here. This is 3 different filters which are in line with your filling procedure. You would see that for the blue and the green line, it needs a certain time reflected then by the filtered volume and also the vials that you potentially fill with that until you reach the target concentration. So by that you could very nicely decide now on how many vials you need either to discard or how you need to optimize your process to be on target from the volume 0. So either select another filter or adjust your filling settings. The implementation of the FlowVPE is on the one hand just, let's say, the technical due diligence, so the feasibility to do the offline comparability with the established gold standard method A280, but it is a bit more like that. So we divided the process in 3 major stages or stage gates. The first is stage gate 1 with the technology due diligence, the equipment interfacing, and also then the lab feasibility, while the stage gate 2 is already looking at potential drug product impact and tox assessment. You can again imagine you now have a recirculation loop with a peristaltic pump and the UV exposure on your drug product, so you need to carefully design lab studies to mimic worst case scenarios if you have a potential drug product impact. In parallel, you do the analytical method development, which needs to be specifically assessed for every NME. You need to ensure a good cleaning and sterility validation. You have to qualify your instrument and then have the aseptic assessment from QA. In the final stage, you then each time decide on the respective NPI. So are you in a concentration range that would be applicable for FlowVPE? You validate this then during your process performance qualification runs. You have to enter that into the regulatory filing since it is a new method to measure protein concentration, and then you enter into the life cycle management. So all that is quite some effort of many different functions, and we are really happy to see the tools spread within J&J, but even also then with outside of J&J, involving all relevant different teams and functions. So now some words on conformational protein stability monitoring via the nanoDSF. Why do we need to monitor conformational stability? I think all of us are aware of the problem of protein aggregates of different sizes, so ranging from the subvisible to the visible range. Sometimes neglected is a bit the initial step that might then boost aggregation over time, which is the protein unfolding. And this protein unfolding is caused by different sorts of stress, which could be mechanical, free thaw, light, just to name a few. It basically can happen during every stage of manufacturing, ranging from the formulation development up to then the fill finish. So we looked for a tool that allows for low volume, high throughput monitoring of this unfolding. So we identified the nanoDSF technology as a very reliable and robust tool to monitor now this protein unfolding. You see here a picture of the very compact instrument basically fitting on every or any lab desk. And the principle of the capillaries with a moving laser detecting the tryptophan and tyrosine fluorescence at 330 and 350 nanometer. The sample volume is 10 microliter, and I think Silvia will also later on a bit more elaborate on some features and added value properties of this technology. In principle, what's happening is that you run a temperature gradient on your sample and measure the fluorescence intensity, by which you then can determine, let's say, the fraction of unfolded versus folded. The half maximal point between the lowest fluorescence intensity, where the protein is still completely folded, and the highest fluorescence intensity, where your protein is then completely unfolded, is called TM, so the melting point, and this is the indicator for the protein stability in the first run, if you want to say. So why is it now? So why do we now get curves like that? So if you look at this little sketch here, so the more you heat up, the more the protein will unfold, and the more of the intrinsic tryptophan and tyrosine will get exposed, and thus emit the fluorescent signal. The less stable a protein is, let's say the earlier it will unfold and the earlier you will get the fluorescence intensity. This is also some example data for a monoclonal antibody that we generated. I quickly guide you through this graph here. So we have here with the light blue line, we have a sample that has been run through the nanoDSF, and has been stored at 5 degrees for 3 months, showing a melting point of 69.1 degrees Celsius. The light brown curve here represents a sample that has been stored at 25 degrees Celsius for 2 months, exhibiting a TM of 68.1. Already a slight shift to the left with also a significant reduction in amplitude. And the third sample has been stored for 3 months at 40 degrees, already showing a reduction of the TM to 66.7. So you see the clear shift to the left, indicating that the red sample is significantly less stable than the blue one, due to the fact that at a lower temperature it's already completely unfolding. So in general we can say that a lower TM corresponds to a less stable protein, and this with as little volume as 10 microliters, so very a nice tool to assess the stability. So we challenged the nanoDSF results with a variety of orthogonal assays. One direct comparator is the FIPA, standing for Fluorescence Intensity Protein Aggregation Assay. This is also an in-house assay that we developed, which gives you as a result the amount of protein aggregates reflected by the increase in fluorescence intensity. But we also used the classical gold standard tools like the MDI, so microflow digital imaging, turbidity, size exclusion, and also let's say others like CAF, CSDS. Here shown just some representative data exactly on the same samples that I already have explained you on the previous slide. In this graph you see in blue bars the amount of aggregates reflected by the FIPA, and with the black squares you see the TM, the representative TM of the nanoDSF. Here we have the different samples again with 5 degree, 25 degree and 40 C. Here we have the same lineup but at a lower concentration. When you now look at the graph, you see again a strong increase in aggregation for the samples that have been incubated for 40 C, and in parallel a significant reduction of the melting point, which is in nice correlation with the aggregation count. The same is true when you then look at the whole orthogonal set of assays. So while the 0 months 5C looks clean and is within spec, which is in agreement with all the tested assays, the 40C 3-month sample is showing an out-of-spec result for basically all the tested assays. So again, reflecting that the nanoDSF is indicative also for the orthogonal platform. Of course, this is now just a representative data set for one selected molecule. We do this testing for basically any molecule that we have in our lab or in our development. And by that, we are in the comfortable situation to really optimize the formulation, but also then the process parameters towards ensuring highest quality of our molecules and products with respect to conformational stability. The next stage of nanoDSF implementation is to spread the technology at all different spots during the supply chain to have a kind of real-time monitoring of stress impact at the different stages, feed all this information into a central database that will allow to predict, forecast, and align the data sets from the different samples and materials. And by that then, let's say, forecast the behavior of new molecular entities that get into the development and supply chain. Quite some people involved obviously in these projects. Today I just really scratched a bit on the surface. There is a lot of work ongoing with these two technologies on several products, very different modalities. And I would really like to thank a lot of people from different functions that supported these projects and brought it to a stage where we can really use it in our daily business, even then in the GMP environment, as it is already true for the FlowVPE. So with that, would like to thank you and hand over to Silvia to now really get into the depths of the nanoDSF features and potential applications. Thank you.
Thanks, Marcel. Great presentation and a wonderful way to get things started, and should be a very nice transition into Silvia's talk. But before we get to that, I just want to remind everyone to send in their questions for our Q&A session, which will be coming up right after the next presentation. Just add your question to the Ask a Question box on the right-hand side of your screen and hit submit. All right, with that, let's get on with our next presentation. Silvia, the floor is yours.
Thank you for the kind introduction and welcome everyone. And thanks of course to Marcel for this very interesting presentation on how you at Janssen use innovative technologies to improve the fill and finish process of biologics. And we've heard that nanoDSF can be an extremely useful addition to the analytics toolbox to monitor protein stability, for example in force degradation studies. The device that runs nanoDSF is the Prometheus. And during the next 10 to 15 minutes, I would like to explain in more detail how the instrument works and show additional examples relevant for the whole protein production workflow. I would like to start with drawing your attention again to the challenges scientists in today's manufacturing are confronted with. To stay competitive in this field, companies need to optimize their manufacturing process. That means ideally having short standing times and short process cycles. So a process cycle, that's a time to complete the whole manufacturing process for a product. At the same time, they not only produce one product, but often need to deal with a larger variety of molecules, which all demand their individual processes and control strategies. That's why they need analytical technologies that are fast, flexible, and work with different types of molecules or sample conditions. We've also heard from Marcel that manufacturing is often performed across multiple sites or partly even outsourced, means that the analytical tools should deliver consistent and comparable results across different sites. And last but not least, large molecule drug products are simply expensive, so there will always be only limited material available for the analytics. The Prometheus can help you tackle all of these challenges, and I would like to present you now exemplary data and applications for that. Before jumping to the application examples, let's go quickly back to what kind of data the Prometheus actually generates. It uses the nanoDSF technology to assess protein stability. nanoDSF stands for Nano Differential Scanning Fluorimetry. And in contrast to classical dye-based DSF, nanoDSF looks at changes in the intrinsic fluorescence of proteins upon protein unfolding. So it doesn't require any modification of the sample. And the fluorescence recorded originates from the amino acids tryptophan and tyrosine. As you can see on the left, these tryptophan residues are often buried inside the hydrophobic core of a protein. And when this protein gets exposed to increasing temperature, it ultimately will lose its three-dimensional structure, and the tryptophans become exposed to the hydrophilic environment, which dramatically changes their fluorescence emission properties. We detect this change in fluorescence and can thereby record unfolding profiles going from a folded to an unfolded state, as shown on the left. And of course, these signals can also look much more complex. The transition midpoint, or the TM or melting point, can be used as a proxy to assess protein thermal stability across different conditions or protein variants. Thermal unfolding profiles are not the only data Prometheus generates. Additionally, it allows you to look at chemical stability of proteins when a denaturant gradient is applied instead of a thermal ramp, and gives you then info on the CM value or the free folding energy delta G. And you can run isothermal experiments or study unfolding and refolding kinetics, as well as aggregation, and the latter providing info, of course, on the colloidal stability of proteins. Here's now our first application example. It comes from upstream development of biosimilars. We teamed up with UGA Biopharma, who wanted to identify the best growth medium for biosimilars production, and we compared the originator antibody with 16 biosimilar candidates. Shown on the left-hand side are thermal unfolding profiles of the candidates and the reference molecule in red. And below the unfolding profiles, you can see the first derivative plot of these signals, and we actually use those to extract the TM at the maximum of the peak. With nanoDSF, we were able to identify a candidate that showed a very similar unfolding profile to the originator, and that's the one in the top middle panel. And that one was also ranked best by conventional HPLC-based methods. And we did this by looking at the whole unfolding profile and not just comparing the melting points. I wanted to mention that here because there are, of course, multiple ways you can look at the data. The nanoDSF experiment took much less time than the chromatography-based method and required less sample. And this shows you how nanoDSF can provide comparably quick answers when throughput or time is an issue. In a production setting, you could, for example, apply it to compare production batches. And let's say if the batch is fine, you can move on to the next step. And if not, you can follow up with orthogonal analytical technologies such as these HPLC-based methods. This way, you wouldn't spend unnecessary time on process analytics and only characterize those samples in depth that require you to do so. Shown on this slide are the results of a study we did in collaboration with Coriolis. We used an antibody buffer screen to compare nanoDSF to microDSC. And microDSC is an alternative and well-established method to analyze protein stability. And we were able to demonstrate that both methods, nanoDSF and microDSC, provide precise and consistent data. The middle panel shows the TM values from both methods, and there are slight differences in the absolute melting points, but the relative correlations and ranking between the different conditions are virtually the same. However, nanoDSF overcomes several drawbacks of microDSC by its innovative capillary format, and I will come to that later in more detail. For those of you working with DSC, the main advantages are the following. Firstly, you need only 10 microliters per sample for nanoDSF. Secondly, the capillaries are single-use and we have no flow cell in the device. That means no equilibration times or washing steps. And it also works in any buffer, so even with detergents, and does not need any sample preparation such as dialysis or filtration upfront. And that's the reason why Prometheus requires significantly less sample and time for such screening projects when compared to microDSC. In protein production, this can be important when you want to, for example, shorten your hold times and process cycles, or when you need to produce a diverse set of molecules. This last example addresses the challenge that analytical technologies in protein production need to deliver precise and consistent results all the time. And some data showing that Prometheus is exactly doing that is presented here. On the left-hand side, you see an overlay of 48 unfolding profiles of streptavidin. So up to 48 samples can be measured in parallel in one run. And we performed this experiment on 10 different devices. We then looked at the variation of the initial ratio and the melting point within a single device and across the instruments. And I think the resulting standard deviations and relative standard deviations are quite convincing. They are extremely low. For example, we have here a variation in the melting point of 0.1 degrees within a device and 0.3 degrees across different devices. And this will allow you to easily compare the results generated at different sites or different process steps. But how does the Prometheus actually work? So how can we analyze protein unfolding with high throughput, low sample consumption, and in a precise manner? This is what is shown here. So the samples are loaded into glass capillaries by dipping them in the sample solution. The capillaries will automatically fill themselves by capillary forces, so there's no need to pipette anything. This also works, by the way, for very viscous samples, which can be important for everyone working with highly concentrated liquid formulations. Each capillary holds 10 microliters and up to 48 can be measured in a single run. We offer two types of devices. The NT48 works with single capillaries and has room for 48 of those. The NT-Plex works with capillary chips, and the capillary chips contain 24 capillaries, which can be all loaded at once by dipping the chip in a 384 well plate, thereby reducing the hands-on time at the device. The capillaries or cap chips are then placed on a thermal element, shown in gray here, that allows heating the sample between 15 and 95 degrees. And with an optional upgrade, you can even go up to 110 degrees. In a typical experiment, one would choose a heating rate of 1 degree per minute from 20 to 95 degrees. So the whole experiment takes about 75 minutes. But you also have the option to select a lower or higher rate, anything between 0.1 degrees per minute to 7 degrees per minute. The capillary tray then moves forward and backward while we read out the fluorescence emission at two wavelengths, 330 nanometers and 350 nanometers. Recording the signal is done on the fly, so the tray doesn't stop in between. And a scan of the whole tray takes about 3 seconds, which means one data point per sample every couple of seconds. And even with a heating rate of 7 degrees per minute, we end up with several hundred data points per unfolding profile. And that's how we achieve such a high data point density and thereby precision and resolution with Prometheus. But why is recording two wavelengths, so 330 nanometers and 350 nanometers, actually totally sufficient to look at protein unfolding? On the left-hand side, you see a typical fluorescence emission spectrum of tryptophan. And as mentioned earlier, in the folded state, these tryptophans are often buried inside the hydrophobic core, and the spectrum shows a maximum at around 330 nanometers. When the protein now unfolds, these tryptophan residues get exposed to the hydrophilic environment, and the emission maximum shifts to around 350 nanometers. That's what we call a redshift, so a shift to longer wavelengths. And additionally, we can see that the fluorescence intensity decreases due to quenching. Some proteins show a blue shift instead of a red shift, so a shift to shorter wavelengths. And this happens when the tryptophans are already surface exposed and the fluorescence is dominated by tyrosines. As you can see here, looking at only these two wavelengths is therefore completely sufficient to pick up changes in the emission spectrum of a protein. In the center, you can see a typical signal of the individual wavelength during a nanoDSF run with a temperature ramp. First of all, we see a decrease in the overall fluorescence intensity due to collisional quenching, but we can see an additional transition corresponding to the unfolding of the protein. However, what yields a much more robust analysis is using the ratio of these two fluorescence intensities, so 350 divided by 330. And what this result looks like is shown in the right panel. We obtain an S-shaped unfolding profile for a red shift and a flipped Z-shape for a blue shift. And the ratio signal is very specific per protein unfolding and independent of quenching effects or the buffer background. And this rapid dual UV detection is actually the reason why the measurement works in any buffer or with any additive. From the derivatives of this ratio signal, shown here in blue, we then calculate the melting point by looking for maxima in this derivative curve. Of course, the single wavelength signals are outputted as well by the software, and you can use those in addition to the ratio signal. As mentioned in the beginning, Prometheus can additionally be employed to measure protein aggregation in parallel to analyzing conformational stability. This is done via our backreflection technology, which is an optional hardware upgrade. It's a scattering-based technique in which we detect loss of reflected light that passed the sample. So the principle is shown here on the right-hand side. When aggregates form, more light is scattered and less light is reflected back to the detector. And this measurement is done in parallel to the fluorescence detection on the same sample at the same time. The signal looks similar to an unfolding profile, and an important parameter that can be extracted from this data is the T-ag, or the onset of aggregation. And this is the temperature you typically want to keep your sample below of because, of course, aggregation is often irreversible. To sum it up, why is Prometheus a great addition to the analytical toolbox in protein production? It offers ultra-high resolution and precision because we only record two wavelengths and thereby achieve an extremely high data point density. You can use it for all types of proteins over a wide range of concentrations, so between 5 micrograms per ml to over 250 mg per ml. No need to dilute or process the sample, and it works in any buffer. You can do fast measurements with a flexible heating rate and measure up to 48 samples in one run, so it's an ideal tool for screening. And for the Prometheus, we also offer software that makes the device ready for 21 CFR Part 11 compliance, which is of course important for everyone working in a regulated environment. I would like to finish my presentation with highlighting that the Prometheus can not only be useful in manufacturing of biologics, but is already applied in earlier steps such as discovery research or characterization. It's therefore an ideal tool to monitor protein stability along the whole biologics workflow. So if you would like to learn more about Prometheus or nanoDSF, please visit our website or get in touch via email. And with that, I would like to thank you for your attention, and I'm happy to answer questions.
Thanks, Marcel and Silvia. Excellent presentations. I think the audience now has a greater appreciation of the importance of protein stability measurements and how the Prometheus can assist in those endeavors.